Hello students, welcome to PG Pathshala. I am Anup Kumar Kapoor from Department of Anthropology, University of Delhi. Today we are going to talk on module Genetics and Human Health from the paper Human Population Genetics. The main objectives of this module is that you will be able to understand the importance of genetics in health, whether monogenic or complex disease, role of genes and genetic variations to modify the status of disease. You would be able to understand the advantages of alternation in genome in human health and also how other factors such as environment, behavior, etc. can modify the genetic effect. And you will also know how a genetic status could be deleterious in one population but advantageous in other conditions in different populations. Now we take the first part. This study module deals with a very little important prospects of life, our genes, which is a region where there is a lot of human diseases domain as per medical aspects. We will have a look for the inheritance patterns, gene function, and inter and intra differences between people and populations. Genetics basically deals with the genetic components and traits passes to offspring to parents. These traits can be height, skin or hair color as well as complex disease susceptibilities. When we combine both clinical phenotypes and a basic science, issues related to medical genetics can be addressed. It makes us understand the function of health and disease in the human. Overall advancement in technologies created opportunities to understand, prevent, treat, and provide therapeutic interventions for the human diseases. For the breast cancer, it is now frequent in reporting about the presence of BRCA1 and BRCA2, the genes, to be used as a causative part and directly linked to the condition and called to have the disease. This is highly misunderstood. Each person has the BRCA1 and BRCA2 genes for their function work. That is, in breast and ovarian cancer, the tumor growth is suppressed. Breast cancer risk is due to inheritance of mutated alleles either from father or mother, which removes that protective shield which suppresses tumor growth as per reported by Narod and Salmena in 2011. Conventionally, single gene single disorder was pronounced, which means diseases which are caused by mutation in single genes such as cystic fibrosis or Huntington disease. Yet at present, there is a paradigm shift from individual genes to multiple genes, that is genomics, and their gene-gene interactions as well as gene-environmental interaction, what we call is epigenetics. Our apprehension of complex genetic disorders such as malignant neoplastic disease and diabetes has been increasing due to technology advancement. These diseases are influenced by a number of risk factors, including environmental stimuli. This has resulted in a more mainstream approach to the application of genomics to a broad diversity of clinical problems in the healthcare context. Ridley has pointed out that the tendency to identify a specific gene as the cause of disease obscures the critical function of genes in human wellness. It has also noted that genetic disorders like beta thalassemia are inherited in autosomal recessive pattern can be checked and removed from the population because both disease alleles are required to cause disease. Carrier marrying carrier may have offspring with both disease allele, which would lead to disease. Now from this figure number 1, we can see the pedigree with autosomal recessive inheritance pattern. In this figure, there is a female who is carrier and the male is also carrier. When they produce the children, there are four varieties of children. The number 1 is the unaffected, they are not a carrier and no disorder. In the second child, they are carrier but no disorder. Indicates this pedigree indicates very clearly. In the same way, in the third child, that is, they are carrier but there is no disorder. But the fourth child, they are affected as a disorder. That shows that this autosomal recessive inheritance pattern is very clear. In human genome catalogs, list of diseases named after the doctor who identified, but the genes are yet to be identified. The diseases caused by gene do not define the genes. It is like defining body organs by diseases. Heart to cause heart attacks, cirrhosis caused by the livers, and kidneys to cause renal failure. But the cataloging is done in similar fashion, which may be misled. It is true that most of the genes we know about the malfunction causing diseases, and this is very small piece of information about genes. An individual has wolf histocon gene doesn't mean that he or she might get the disease, which is incorrect information. Subjects who had wolf histocon syndrome doesn't have wolf histone gene, but all other individuals have. Their disease condition is because of missing of particular gene, but it is present in all other normal individuals. It implies that diseased person had a mutation in particular gene, not just had a gene, as per Ridley 1999. Classification of genetic disorders is done in a number of ways, as explained by Trent in 1997. For the topic undertaken, it can be classified as the first, monogenic, that is single gene disorders, the second, polygenic or multi-gene disorders, and the third one is multifactorial disorders. In addition, there are chromosomal disorders such as Down syndrome, as reported by Cohen 2003, and somatic cell disorders such as cancer, in which the genetic abnormality was not present at conception but was acquired during life and is found only in specific cells rather than in all cells of the body, as reported by Frank 2009. Table 1 below lists a number of more common genetic disorders. The table lists of common genetic diseases and chromosome. Here we will see in table number 1, on the left side the disorders are there and the number of chromosomes, on which chromosome this gene has been identified. For example, the Angelman syndrome, it is on the 15th chromosome. The second is Canavan disease, that is on 17p. Then we have Cry-du-Chat, that is on chromosome number 5. Then cytosis fibrosis, that is on the chromosome 7q. Then we have Down syndrome, that is chromosome 21. Then we have hemophilia, that is again X chromosome. Then we have Klinefelter syndrome, again it is on the X chromosome. Then we have phenylketonuria, that is again on 12q. Then we have sickle cell disease, that is on the chromosome 11p. And then we can see the Turner syndrome, that is again on X, that is the sex chromosome. Monogenic disorders, when a mutation in one or both alleles of a single gene is the causing factor in a genetic disorder known as monogenic disorders. Studies on monogenic disorders like Huntington's disease, that is known as HD, made us realize the impact of genetics in human health. Disease SD is a relatively rare disease but is caused by genetic abnormalities. Huntington's disease is an phenotype of genetics. the changes in environmental aspects like healthy lifestyle, medicine, can modify your lifestyle disease like diabetes, obesity, but pure genetic disorders like beta thalassemia, HD are inevitable and cannot be reversed if you had genetic abnormalities. For the disease, your genes decide your future, as reported by Ridley in one of his work in 1999. The heredity of monogenic disorders is an important consideration in developing a policy framework for the protection of human genetic samples and information. Monogenic disorders allow relatively accurate inferences to be drawn about a person's future health from their current genetic status. However, this is not the case when the vast majority of these genetic disorders, where the relationship between genetic status and disease is highly complex and contingent. Now, from this figure, that is monogenic versus complex disease, when we see at the monogenic disorders, mutation is taking place on a single gene, whereas in complex disorders, many genes are there, gene A, B, C, D, right? And then when we study the impact of mutation or disease phenotype, and then we see the genetic risks in different families, it comes to almost the same. Whereas in the complex disorder, it is very interesting the influence of variations in different genes on disease phenotypes. The life of environment or the lifestyle is very clear that genetic risks in different families. Then we see polygenic disorders and haplotyping. The vast majority of medical conditions with genetic perspective involve either the interaction of a number of genes, that is polygenic, or the interaction between genes and the environment, that is multifactorial disorders, which is very complex to understand fully, as reported by Trent in 1997. As Ridley in 1999 has stated, unless you are unlucky enough to have a rare and serious genetic condition, and most of us do not, the impact of genes upon our life is a gradual, partial, blended sort of thing. You are not tall or dwarf like Mendel's pea plants, you are somewhere in between. You are not wrinkled or smooth, but somewhere in between. This comes as no great surprise because just as we know it is unhelpful to think of water as a lot of little billiard balls called atoms, so it is unhelpful to think of our bodies as the product of single discrete genes. According to Ridley, 1999. Total of 14,014 genes have been mapped and reported to individual chromosomes, and out of which 1,639 genes are associated with genetic disorders, as suggested in Human Genome Database published in 2002, as per Kent et al. 2002. The rate of discovery has slowed dramatically regardless of better technology on the mark. It may be that most of the simple linkages have already been made. Only 17 genes were identified with a genetic disorder out of the last 3,783 genes which had mapped. According to Francis Collins, Director, United States National Human Genome Research Institute, known as NHGRI, after success of the Human Genome Project, the next whose thing is to produce a human haplotype map. A number of closely linked alleles along a region of a chromosome which tend to be inherited together are referred as haplotypes or haplotype blocks. According to NHGRI, that is National Human Genome Research Institute, the elucidation of the entire human genome has made possible our current effort to develop a haplotype map of the genome. The haplotype map or HapMap will be a reference work that catalogs the genetic variations are most important to health and disease. All individuals have 99.9 identical DNA sequences. Individuals' disease risks may be greatly affected by the variations inherited or occurred. Single base differences in the DNA sequence where individuals differ are called single nucleotide polymorphism. In the short, it is called as SNPS. Group of SNPs in close proximity on the same chromosome are inherited in blocks. The pattern derived from SNP group is considered as haplotype. There could be a large number of SNPs in the blocks, but a relatively few SNPs are enough to uniquely identify a haplotype. Mapping of these haplotyping blocks, including the specific SNPs that identify blocks, is known as HapMap. Now from figure 3, we can see the set of SNPs inherited together as a block known as haplotypes. Now here we can see there are 6 combinations are there, and with the NP single nucleotide polymorphism, that is AG is coming, forming AGT, GTA and AGA haplotypes, a set of closely linked genetic markers present on one chromosome which tend to be inherited together. This is the way how the genes on one chromosome are inherited that has been shown. Individuals at risk for particular disease could be identified by comparing the patient's genetic pattern with the known patterns from HapMap. It will help researchers to locate the diseases associated blocks or genes. The HapMap will enable researchers to examine drug efficacy to provide specific treatment because people with the same disease may respond differently to the same drug treatments. Finally, haplotype mapping will disclose the role of disparity in individual responses to environmental factors. Recent research reveals that there are a number of common haplotypes, although theoretically, in a chromosome region, there could be a large number of haplotypes. There are 4 or 5 common patterns across all populations, which would make researchers to minimize their experiments by testing the genetic predisposition block by block, node by letter, for such complex diseases as cancer, diabetes, hypertension, and Alzheimer's, as reported by Gabriel et al. 2002. Common variant hypotheses proposed by researchers have made 3 important points. First, the human genome can be objectively divided into straightforward haplotype blocks, each ranging 11,000 to 22,000 DNA letters, but only 4 to 5 different variations in the letters. Second, the haplotype blocks are comparable across individuals from Asia, Africa, Europe, indicates that MAPHAP will have broad effectiveness for most people. Third, that 90% of genetic variation in a region of the human genome was able to be captured by the haplotype blocks, as reported by Morton in 2002. This type of study is an important step towards developing a more powerful statistical approach to study complex human disease. In complex disease, genetics could not be able to understand the mechanism biology, even though great effort has been put during the last 10 to 15 years. From our efforts, we have come to comprehend that complex diseases are not caused by a single high penetrance gene, but from more unproteus risk factors common in populations such as Mark Dallas, one of the authors. This study provides an opportunity for scientists to treat and improve models of population biology and molecular evolution. For initials for population, genetics work out easy, but for a disease, it is comprehensive and difficult task to handle, according to Morton 2002. Multifactorial disease and environmental factors. Multifactorial disorders means not only mutation inherited will make individual vulnerable or predisposed to develop the condition, but there are other risk factors such as unbalanced diet or stress or exposure to some environmental factors which are involved to contribute in the expression of the disorder. Common health problems in humans such as heart disease, hypertension, psychiatric illness such as schizophrenia, dementia, diabetes, cancer are multifactorial. For a decade, the prevailing convention was that nurture, that is the environment, is more important than nature, that is the genes. In course of human health development as reported by Rose et al. 1984. The rate and burden of genetic research in recent times, however, have been in other direction prior to convention, that is perhaps too far in the direction of genetic exceptionalism and determinism. In fact, the depiction is far more compound. An individual is not the addition of a column of traits and behaviors resulted by individual genes. As an alternative, it was proposed that individuals have the total product of genes, the complex interaction of those genes, and the comprehensive interaction between that genetics and environmental factors is present. Even a straightforward situation to the environment minimize the dynamic and widespread nature of this relationship. If we had a look for environment quality, a balanced and hygienic diet, good health care assess. Exercise will allow the entire expression of genetically inherited traits such as height. Over a time span, other features of the physical environment also will modify human health and development, like air and water pollution, endemic disease, drought, and war. Choice and chance also play a significant role, that is, smoking and skydiving create dangers to health unrelated to genetic inheritance, and a high speed head on car accident will always trump good genes. It is well said that you had better get used to such indeterminacy. The more we delve into the genome, the less fatalistic it will seem. Gray indeterminacy, variable causality, and vague predisposition are the hallmarks of the system because simplicity piled upon simplicity creates complexity. The genome is as complicated and indeterminate as ordinary life because it is ordinary life. This should come as a relief. Simple determinism, whether of the genetic or environmental kind, is depressing prospect for those with a fondness for free will. The environment is also occupied of social entities that have an effect on our health and the chances to acquire our complete potential. For example, if a woman is restricted to receive higher education, or if community through discrimination blocks certain racial groups from any employment, then utilization of essential intellectual ability will be minimum. Now we see from figure the flow chart showing the interaction of genetics, behavior, and environment resulting health status. Now we have the health in the center point, that is the main focus. Then we have C genetics responsible for the behavior. Behavior is responsible by the environment. Environment also by the genetics. And on the other side, on the outer side, it is epigenome, proteome, regulome. All these genome, they have been described how they are acting, the interaction of all these dynamics with reference to genetics, behavior, and environment. Disease or protective trait. Genetic variations or mutations are commonly presented as diseases or disorders, but in the past, some of these mutations considered to enhance the prospects of survival in some environmental context. Some example is which the genetic abnormality does not cause any disease condition for carrier, that is inherited affected mutation from single parent, involve autosomal recessive conditions but would result in disease condition if a child who had both inherited genes affected from the both parents, again reported by Trent in 1997. Stop. Now we have the example from beta thalassemia, is a common blood disorder in the Mediterranean region and in several parts of Southeast Asia. The genetic abnormalities in beta-globin gene causes disruption in the synthesis of beta-globin protein, which are found in RBCs. Carriers who have only one allele is mutated are expected to have mild anemia that does not cause any serious health problems. But both the allele affected person has severe anemia, which usually requires lifelong blood transfusion, whereas the carrier state found in as many as 1 in 10 people in some populations get protected against malaria because RBCs of carriers individuals are small and pale, which does not make it available a good environment for malaria parasite in which they can grow, as reported by Galeno and Orziza in 2010 as well as by Rund et al. 1991. Then we have another example that THS disease, that is TSD. It is common in the Central and Eastern European, basically in the Ashkenazi Jewish community, then from Middle Eastern, that is non-Jews or Sephardic Jews, which carry a frequency of approximately 1 in 30. THS disease is neurological degenerative disease which results in death in 4 to 5 years of age. Similar to beta thalassemia, there is no symptoms of the disease for TSD carriers. But it has been observed that this carrier state had protection against tuberculosis for Jewish population who lived in the restricted conditions of the ghettos for the time past, as reported by Rapi et al. In 1988. The other example is cystic fibrosis, CF, is common among Caucasians with the prevalence of 1 in 25 of whom are carriers for CF, also found in numerous ethnic groups. The chloride movement gets affected and found across the cells and causes lungs and pancreas problems in disease state, as reported by Neodan et al. 1989. Carriers for CF do chloride, that is salt, movement was not found across their membranes, similar to those of non-carriers, and are at reduced rate of mortality from diarrhea. When cholera and dysentery were endemic, carrier of this mutation may become beneficial over the many thousands of years of evolution. Carriers commonly do not have the symptoms of CF. Therefore, carrier status can be found through DNA screening, but the inheritance of CF mutated allele from both parents may lead to severe health problems, although CFS is heterogeneous in terms of severity, as reported by Workman et al. 2006. Now we take sickle cell anemia is a common genetic disorder among individuals from Africa and the Mediterranean area. It is caused by mutation in the hemoglobin gene. The carrier state affords protection against malaria. However, because carriers have abnormal red blood cells that die soon after being infected with the malaria parasite, compared with the normal red blood cells, which continue to work and to provide an environment in which the malaria parasite can grow. In an evolutionary sense, being a carrier for sickle cell disease is a good thing if one lives in a region in which there is endemic malaria, as reported by Lizzato 2012. The link between an individual's genetic status and the expression of a genetic disorder, and the link between the expression of a disorder and particular health outcomes, A, need to be understood to address disease status and therapeutic interventions. In particular, there are difficulties in interpreting genetic information in a way that is reliable and relevant for many contexts in which genetic information is or may be used now and in the future. Now the question comes, what are the issues in genetics and health? Recent times, genomic research may significantly change the practice of healthcare and management, but only genomic research is not sufficient to relate this upcoming field to improve human health. we need to carefully understand all ethical, legal, and social issues raised by this type of research. Such study is vital to being able to use genomic research to help patients and preventing misuse of new genetic technologies and information available. Issues raised by genomic research include ethical standards required to work on human subjects or tissues, possible discrimination by employers and health insurers, and consideration of social, cultural, and religious perspectives of genetics and health. There is a need of proper regulation of genetic testing, and privacy of patients should always be maintained to avoid any discrimination as reported by WHO in 1997. Now we summarize this module, that is, the genetics basically deals with the genetic components and traits passes to offsprings to parents. These traits can be height, skin, or hair color, as well as complex disease susceptibilities. When we combine both clinical phenotype and a basic science, issues related to medical genetics can be addressed. Conventionally, single gene single disorder was pronounced, which means diseases which are caused by mutation in a single gene, such as cytofibrosis or Huntington's disease, etc. Yet at present, there is a paradigm switch from individual genes to multiple genes, that is genomics, and their gene-gene interactions as well as gene-environment interaction, what we call is epigenetics. Classification of medical conditions or diseases linked to genes is done in a number of ways. It can be best classified as monogenic, that is single gene disorders, or polygenic or multigene disorders, and multifactorial disorders. When a mutation in one or both alleles of a single gene is causing factor in a genetic disorder is known as monogenic disorders like Huntington's disease, beta thalassemia. Polygenic disorders, medical conditions with genetic perspective involves either the interaction of a number of genes that becomes polygenic. Then individuals at risk for particular diseases could be identified by comparing the patient's genetic pattern with the known patterns from HAP-PAP. It will help researchers to locate the diseases associated blocks or genes. Multifactorial disorders means that a mutated allele inherited for particular conditions implies that the individual is vulnerable or predisposed to develop the condition. Other factors such as diet or stress or exposure to certain environmental factors are involved to ensure the expression of the disease or conditions. Common health problems in humans, such as heart disease, hypertension, psychiatric illness such as schizophrenia, dementia, diabetes, cancer, are multifactorial. Genetic variations or mutations are commonly presented as disease or disorders, but in the past, some of these mutations considered to enhance the prospects of survival in certain environmental contexts, like carrier state of beta thalassemia is protected against malaria. Issues raised by genomic research include ethical standards required to work on human subjects or tissues, possible discrimination by employers and health insurers, and consideration of social, cultural, and religious perspectives of genetics and health. All these issues are need to be addressed through proper management. Thank you.